Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been.
Response. The immune system protects the body against invaders, such as bacteria and viruses. The skin serves as an important first line of defense against foreign invaders. The breaching of the skin, for example, by a splinter, could provide a route for bacteria to enter the body. These invading bacteria are regarded as non-self substances by the body's immune system, thus triggering an inflammatory response in an attempt to destroy the invading agents. A special type of WBC, known as a macrophage, engulfs the non-self bacterium, reducing it to tiny remnants known as antigens, which are displayed on the external surface of the macrophage, thus serving as an antigen-presenting cell or APC. This alerts the body's other macrophages to the presence of bacteria in the body. The macrophage also communicates with these cells by releasing cytokines, which fit snugly into the receptors of the cells, much like a lock-and-key mechanism. This results in the increase in the number of macrophages at the site of the inflammation. Additionally, some of the normal cells also function as antigen-presenting cells by ingesting some of the bacteria and displaying the antigens on their surface. This type of immune response is immediate and non-specific and is known as innate immunity. However, as the macrophages cannot destroy all the invading bacteria in the body, they proceed to the peripheral lymphoid tissues, such as the lymph nodes, in order to alert other immune cells to the inflammatory response taking place in the body. As lymph nodes function as a gathering place for several of the cells of the immune system, the macrophages are able to activate the T and B cells, which are different types of WBCs, which are then able to mount an immune response. This type of immune response is specific and as it takes the body a longer time to mount such a response, it is known as acquired or adaptive immunity. T helper cells are stimulated by interleukin-1, secreted by the antigen-presenting macrophages, and in turn secrete interleukin-2, which help in the activation of other immune cells, especially cytotoxic T cells and B cells. Cytotoxic T cells bind to the antigen on the antigen-presenting cells and secrete chemicals which in turn destroy these cells. This type of adaptive immunity involving the cytotoxic T cells is known as cell-mediated immunity. Activated B cells could be of two types: plasma cells or memory B cells. Plasma cells secrete antibodies that bind to the antigens on the surfaces of the antigen-presenting cells or bacteria, which are then destroyed by the macrophages. This type of adaptive immunity involving the plasma cells and antibodies is known as humoral immunity. The memory B cells, along with the memory T cells, survive for several decades and mount a secondary immune response to a future infection by the same pathogen. This same principle is employed during the vaccination of children and adults against common infectious pathogens.